Cortical interneurons and neurovascular coupling. As many of you before stressed, one of our big aims is to understand how neuronal activity and changes, for example, in bold contrast, are intercorrelated. And one important step to understand this correlation is to have a detailed mechanistic understanding of the processes that are in between our macroscopic hemodynamic observable and the neuronal activity. And this is neurovascular coupling leading to dilation or constriction of the vasculature, as well as changes in tissue oxygenation. And it would be really helpful to figure out if there are any kind of hemodynamic signatures of different types of neuron populations. And I will talk about the left part of this problem, and this is our approach. We perform direct measurements of vascular responses using two-photon microscopy, and we interrogate the system by modulating the messenger pathways which we think are involved in this system, and we use optogenetics for cell type specific modulation of activity. And first, a brief introduction on the two-photon measurements. So we have mice with a craniotomy. We inject the intravascular fluorophore, for example, FITC, and then we look at single diving arterioles, how they dilate or constrict in response to a stimulus. And we can do this either in line scan or frame scan mode. We use mice which are either awake or under anesthesia, and this is basically what you get. If you stimulate the mouse, for example, with an air puff which deflects the whiskers, you have a dilation, you have a constriction. Depending on where you look at, it looks a little bit different, but that's the general pattern. And if you look at many vessel segments over a lot of cortical depths, you see an interesting behavior which is not really understood. You can see that the dilation occurs first in deepest cortical layers and at last close to the surface. And in order to collect this data, it was necessary to be able to image at depths between 600 and 900 micrometers. And this was possible by using redshifted fluorophores. It has been proposed or shown already in 2009 that we can image up to 1.5 millimeter cortical depth with that. We are not that ambitious, but I show you here a comparison between FITC dextran, which you may know has an emission of about 520 nanometers in maximum, and Alexa 680, which emits about 200 nanometers closer or far to the red. And this is the same preparation imaged with FITC dextrans. You can see a loss of contrast at about 500 micrometers. And the same preparation imaged with Alexa 680 gains you a couple of 100 micrometers more of imaging depth, which allows you to do functional studies like this throughout a large depth cortex. But back to neurovascular coupling. In recent years, there were many studies which proposed that neurons and maybe astrocytes release messengers which target cells of the vasculature and cause dilation or constriction in relative fast temporal scales. And studies identified that pyramidal neurons in the cortex release prostaglandins and related molecules, and cortical inhibitory interneurons, or short INs, release, for example, neuropeptides like neuropeptide Y, somatostatin, vasoactive intestinal peptide, as well as nitric oxide. And it has also been proposed that neurotransmitters themselves may have a fast effect on the vasculature, as well as extracellular ion levels. And I will focus now on the specific contribution of cortical inhibitory interneurons. And the first basically proof to show that these neurons can cause a vascular effect was to take a channelrhodopsin expressing mouse line, which expresses channelrhodopsin specifically in all interneurons. Channelrhodopsin is a cation channel which can be gated by light, meaning we have now a mouse which expresses channelrhodopsin in all cortical interneurons. And if we shine blue light onto the cortex of the mouse, we activate these neurons. And this is how it's done. So we have a laser light coming through the objective. It illuminates an area of about 230 micrometers in diameter. And when we look at the vascular response to this optogenetic stimulus, which lasts about 200 milliseconds we see that the response profile in its general shape is very much similar to the sensory response. And if you want to see more about a very detailed analysis of this model, you can look into a paper which was published last year by our group. But this was a very good starting point to look further into these inhibitory interneurons. And the first thing one has to appreciate is that interneurons are highly heterogeneous population. You can see this already if you look at the different morphologies, but as well as the electrical properties, the connectivity patterns, and more recently, genetic lineage and marker expression. And for now, I just want to focus on the marker expression, because with that, we can subdivide the interneurons into three major groups, as I try to show here, which express some of these neuropeptides in a non-overlapping manner, like here down VIP or somatostatin and neuropeptide Y. So this already divides these interneurons into non-overlapping groups with different types of proposed vasoactive molecules. Other vasoactive messengers like nitric oxide are shared between them. So this is an additional complicacy, but for now, this is a very good starting point to subdivide them. And our approach now was either to use a pharmacological blockade of specific messenger pathways, for example of NPY or VIP, or to perform an optogenetic activation not of all interneurons but of specific subtypes. And first I would like to show you our results we got with pharmacological experiments. So we blocked VIP receptors and looked at the vascular response to sensory and optogenetic stimulation in the mouse, which expresses channelrhodopsin in all interneurons. And you can see a reduction of the dilation amplitude in both cases. But if you look closely, the onset is the same and the dilation is not gone completely, which raises the question which other vasodilators were involved. And so far we tested acetylcholine and CCK by adding additional blockers, which didn't produce any additional reduction of dilation. On the opposite, by blocking NPY receptor 1, we got a very clear result, which is a complete loss of the constriction in both cases. So you can see this is the normal response, and upon NPY blockade, you see that the constriction is gone. And this data was already published in the 2016 paper. But this led us to ask whether or how the actual response to optogenetic stimulation of specifically this population of neurons is, and if it's maybe only constriction. And for that purpose, we used some more advanced mouse genetics to create a mouse which expresses channelrhodopsin now specifically in NPY positive interneurons. And this is an immunostaining of the cortex slice of this mouse. And you see some of these neurons are now positive for channelrhodopsin. And I want to stress that this is a relative sparse population of neurons, especially if you compare that to the mouse which expresses channelrhodopsin in all interneurons. And then we looked at the optogenetic response side by side between these two mouse lines, and it was kind of a surprise. We have a very sparse population of neurons, but we have a relatively strong response which also involves dilation. And this was our most recent result, and we want to follow up with that, and I will tell you a little bit about that on the last slide. So we conclude we can do vascular imaging of to about 1 millimeter depth with redshifted fluorophores. We have seen that optogenetic stimulation of all interneurons elicits a response which shares key features with the sensory response. We have seen that VIP blockade reduces but not abolishes the dilation upon stimulation, And NPY seems to be the major constrictive messenger, but optogenetic stimulation of NPY positive interneurons causes also a strong biphasic vascular response. This raises the question which other EN interneuron-mediated dilatory mechanisms are in play. Is it maybe nitric oxide or extracellular potassium? And then it's of course interesting to find out whether the strength of this vascular response by NPY positive interneurons is something really specific to these interneurons. So are they very strong drivers of vascular responses, or is this inherent also to other interneuron populations? And we also want to look into the structural relationship between sources and targets, like NPY or VIP vesicles, and the receptors on the vasculature, which may help to answer the question why the dilation occurs first in deep cortical layers. With that, I would like to conclude and acknowledge Anna and all current and former members of her laboratory, as well as Herbert Herzog for providing us the Creel line for NPY expression, and Stephen Adams from Roger Tsien's lab for the conjugation, help with conjugation of the LA680. Thank you.